Good morning. I'm Gula Lifshitz, biotech analyst at Chardan, and it's now my pleasure to introduce Dr. Lalo Flores, President and CEO, and Dr. Greg Rosati, CTO of Century Therapeutics. The format for this session is approximately 25-minute fireside chat, but we'll start off with some high-level overview slides of Century and its manufacturing capabilities before we move on to the Q&A portion. If you have any questions during the session, feel free to type them into the question box in the Zoom chat, and we'll see if we can try to ask them on your behalf. Century team, please begin with your presentation.
Thank you, Gula, for the invitation. A real pleasure to be here with all of you and share our perspective with this audience. So we have a couple of slides. If the second one, if we can go to the second one. So our forward-looking statements, and then if we can go to the next one. I would like to start with this just to level set and remind the audience about Century's vision. For us, since our inception of the company, our vision and ambition is to really become a leading developer of allogeneic cell therapies for cancer. A decision that we made early on was that we were going to commit all of our efforts to develop a cell therapy platform that was going to be based on iPSC technology rather than donor-derived cells. And we're going to spend time explaining why we think that iPSC is the future in allogeneic cell therapy. And when thinking about the platform, we are committing to develop two cell types, NK cells and T cells. But beyond that, which iPSC really reflects to the starting cell, our company, our commitment is all along to complement that iPSC technology with the state-of-the-art cell engineering platforms. But not only is precision gene editing and protein engineering, but very importantly also built-in gene edits into our cells that will allow them to resist host versus graft rejection, which we think is very important. And we can expand on that during the Q&A. But the fourth piece of our strategy from day one, and very relevant to the discussion today, is that we believe that having access and developing a state-of-the-art manufacturing is central to achieving our goal. And so this is what we are all about. And we think we're very excited with the progress we've made today, really putting together what we think is the next generation, one of the most comprehensive cell therapy platforms that include all of these capabilities that really allows our team to have the end-to-end capabilities that are needed to achieve our mission. And so now we're very excited to be able to advance our first product later this year into the clinic. So if we can go to the next slide, very high level, I think Greg will expand on this. This is a high level overview of our platform. Again, in contrast, there are three major components: reprogramming, cell engineering, and manufacturing. We're going to spend most of the time talking about the manufacturing today, but just to remind the audience that everything begins when we reprogram a somatic cell. These are the three cells shown at the left that we use. And we reprogram them so that we can make an iPSC bank. And importantly, this is done only once in the whole process. Once you have an iPSC line that works, that has been validated, then we are able to incorporate multiple genetic modifications that we believe are needed to improve the functionality of our cells. Importantly, because of these cells have a limited replication capacity, we can do this in a sequential manner using precision gene editing that relies on homologous directed repair, a process that allows our scientists to knock out genes while simultaneously knocking in a transgene of interest, a very precise site on the genome. And again, also allow us to perform very sensitive analytical tests to ensure genomic integrity is maintained throughout this process. And finally, we screen many hundreds or even thousands individual clones to come to what we call and manufacture what we call the master cell bank. That is the beginning of the manufacturing process. And then we'll expand. Greg is going to expand about how that is done in our platform. And with that, and again, one thing just to finish is that, of course, manufacturing can be done in multiple rounds. The master cell bank supports the overall life cycle of the process. But everything that happened before the master cell bank is done only once, and that's a key advantage compared to a donor-derived process. Of course, each product will have its own master cell bank because it's the engineering that is introduced to the cells is fit for purpose, right? To enable the right functionality for cell to work in a particular indication. And with that, let me stop and turn it over to Greg.
Thanks, Lala. So just to expound on Lala's point about how one master cell bank for product and how we can use that master cell bank again and again to make our product. This schematic indicates what a process may look like. This is our NK process, and you can see the great proliferative potential of the iPSCs allowed us to get a master cell bank that was quite large. In fact, there were 200 population doubling levels typically from the single cell clone that we select with the proper gene edits to get to that master cell bank. From there, we differentiate the iPSCs into hematopoietic stem cells, HPCs, make a bank there as well, or an intermediate, about 12 PDLs to get there. And from there, we have multiple vials of this HPC intermediate from which we can make multiple NK drug product batches, or in the case of T cells, T cell drug product batches. And you just get a sense here of the number of PDLs, population doubling levels. And an important point to raise here is that every single product, every single batch will have the same number of population doubling levels from that initial clone or from the master cell bank. So very consistent product and processing this way. This ensures high product quality and safety. And because of this, we can scale very easily. We can make batches as large as we like, which I'll talk about in a second. That'll help bring down the cost of goods. And we really believe the fitness of the cells will be consistent. And we've proven that with this many population doublings, of this many passages in these kinds of processes, we have the cells that are fit for purpose. So two major areas we focus on are having a good understanding of our assays and processes, because we want to have consistent product quality. So what that means is we have to understand what makes our product work, understand the key clinical quality attributes, and then choose meaningful assays which will allow us to measure those reliably. If we can measure them reliably, then we know we're making the right product. And ultimately, you have to characterize those assays very comprehensively, because ultimately, if your products get licensed, which we fully intend ours to get there, we have to have assay validation. So the work we do along the way will better prepare us for assay validation, but most importantly, along the way, it helps for high quality products. Similarly, with the process, it's very important that we understand how the process affects the product. Ultimately, the goal is to validate the process for commercialization, but along the way, again, understanding what inputs affect the key outputs will allow us to have better control of our process. All of this is going to lead to better tech transfers, more confidence when we scale our process up or make process changes or facility changes. And as we have problems, which you inherently have in these kinds of very complex processes, we can troubleshoot them faster and basically have better product quality along the way. And then the other piece I just want to highlight is an investment we made in in-house manufacturing, but also a partnership we have with Fujifilm Cellular Dynamics to do manufacturing with them. And what this gives us is two facilities at which to manufacture our products. But why our own? Why not a CMO? So we realized that in-house manufacturing is incredibly critical for complex types of therapies like cell therapies. There's so much institutional knowledge that we're going to build and gain, and we really want to protect that from competitors that you might have through CMOs. And you also want to gain that knowledge yourself and learn along the way yourself with your own people. This will help us iterate better, faster, have better success, be able to make those kinds of changes, and ultimately have better control of our schedule and be able to prioritize our own activities however we want to do it in our own facilities. So about our two facilities, well, one is ours, one is our partners, FCDI's, who is making the first product that will go into the clinic, Century 101. We're making that with FCDI in their facility in Wisconsin. And then our facility in Branchburg, New Jersey is coming online this year. It's a 53,000 square foot facility, multiple suites, capable of making multiple product types, including NK and T cells, as well as other immune cell types. So we're really excited to have these capabilities. And with that, I think we are going to close and happy to take questions and have the chat.
Perfect. So thanks for that overview. You touched on a lot of points that I'd like to expand on a little bit more in this discussion. So maybe to start with the last point that you talked about with regards to the the manufacturing facility. Can you expand a little bit more on the strategic approach there with regard to why you decided to pursue both the internal and the partnered, and also how you envision those capabilities evolving in the coming years as you advance your programs through the clinic?
Maybe I can start. We can tag team with Greg. Look, it was really important for us to establish a strategic partnership at the very beginning. It was, it's very hard to develop, right away your own manufacturing. And FCDI really it's not a CDMO, really it's a true partner to us. So that gave us a tremendous jump start and access to a lot of know-how and talent, and that they were familiar with the early process that we were using for converting iPSCs into immune effector cells. And so that made a lot of sense. And as Greg said, that gave us optionality, having, we will continue to have access to that dedicated access at FCDI facilities, while also now starting to bring online our own. So it was really a strategic decision, was essential to get the company going. And, Greg can expand on that, of course.
Yeah, thanks, Lalo. I think that, obviously you have to start somewhere, and it's oftentimes there's not enough time to start on your own. But when you think about it, as I mentioned, there's a lot of reasons to have your own type of facility for this kind of therapy. And I won't reiterate those. You heard those in the presentation. But I do want to mention, I guess, the three things you have to have your own facility. And we were fortunate to have all three. One is time. So after the first product, we had time to build our own facility. The second is money. It's very expensive to build a facility like this. So many companies are not well funded. We are, so we're very fortunate. But the third is expertise. You need to know how to build one of these and then how to run one of these. So we were fortunate to have a team already in place where we've hired all the key leaders now from facilities, manufacturing, quality, process technology, analytical technology, and a lot of the personnel as well that have run facilities like this, both clinical and commercial, but mainly we're focused on clinical right now. And we know how to do this. We've done it before, and we're going to be very successful at this. And I don't think everybody can say that. You really need to know how to run one of these. So we're very excited to have this opportunity, and we think it's going to bring us a lot of success in our programs going forward. And to have the two facilities for optionality is a really, really big bonus as well.
Great. That makes a lot of sense. And so now maybe let's turn to your process. So you gave that overview slide of the process going from the iPSC to the master cell bank, to the genetic engineering components, et cetera. So can you expand a little on how that master cell bank works within your manufacturing process, and also how that common progenitor line is deployed throughout your pipeline, and kind of the logistics a little bit around that?
So maybe I can start with the common progenitor, so the common progenitor is a very important strategic priority for us, because it would really, when you invest and you have the common progenitor that has the core genetics, as I said earlier, alloevation, for example, technology requires a minimum of 3 dedicated genetics that are going to be maintained across all our whole product line, right? So we also aspire to incorporate in the future some other core modifications. Without expanding into those today, by the way, we will be providing a detailed update on our common progenitor at our June R&D day. So I invite everybody interested in hearing more about the common progenitor and all the core edits to that event in June 13. And so, but then after that, the key advantage of the common progenitor bank is that then, we can just have, you need a minimal set of engineering step. The minimum would be just one additional step to incorporate the car of interest, right? That would be dictate what kind of antigen the drug product will recognize, will be target for killing. And that will really set the stage and identify the product utility, what indication will be used for, right? So really that dramatically shortens the timeline. But then, and then maybe, Greg, you can expand us to the scale of the two, the common progenitor versus master cell bank, and the interplay between the two.
Sure. So scale really comes down to the proliferative capacity of these cells. We can make these as large as we want. or we could expand them as much as we want. And it just becomes a manufacturing strategy you put together to predict, okay, well, how many doses do I think I need to make of a given product over the lifetime of that product? How long do I want my manufacturing process to be? And how much time do I want to invest up front expanding cells to make a common progenitor or master cell bank? And then you just do the math and decide where to stop each one, how many PDLs go into each one. So common progenitor, as Lala said, will feed multiple programs. So there might be a common progenitor for NK, a separate common progenitor for T cell. And then the beauty of that, as Lala said, is you have speed to your next product because now there's only one edit to make, or limited edits versus all the edits. So then once you make that last edit and you make your master cell bank for a given product, that master cell bank is there for the lifetime of the product. You have these vials and freezers, probably across multiple sites because you want to mitigate against risks, right? And then whenever you want to make a batch, you thaw a vial. And we also have this intermediate HPC, which is not really a bank, it's more of an intermediate, but it is multiple vials frozen down. So again, we can make multiple batches from that. And if you do the math on these things, and if you want to add a working cell bank in between, you could. We don't need to because the way the master cell bank is so large, and at least we don't need to for our first product. We've done predictions on size of product needs and batches and whatnot. But in the future, if we wanted to add a working cell bank in there and have another hold step, basically to allow the cells to expand even further, we have data that says we can even expand these beyond the 220 population doublings that I showed in that one slide. So it's all a matter of just doing a mathematical exercise, getting data to prove that you can expand the cells that much, the cells are still fit. And then after that, it's just consistency because you're doing the same thing over and over.
Got it. And you alluded to scaling and COGS advantages. Can you expand on the potential advantages for cost of production and scale and what exactly that means? And in particular, for example, in comparison to some of the other allogeneic approaches that are in development?
Greg, you want to? Yeah.
Sure, happy to. So I think the real advantage is that, and I'll just talk about donor-derived in general. With donor-derived, everybody knows you're going to have to go back to a donor on a regular basis because with donor-derived, you're starting with primary T cells. So the proliferative capacity is limited, right? It's something, it's better than autologous, but it's limited. So even if you can make 100 doses per donor, 200 doses, pick whatever number you want, the largest batch you could ever make is the batch that makes that many of those doses per donor, the maximum number of doses per donor. And it's the largest batch you could ever make. With us, we can make whatever batch size we want. Now, we're not going to get crazy and make all the batches for the lifetime of product up front. That would be a poor investment. But you'd think about the stage you're in. And phase one is relatively easy to make large enough batches. And you might make a few, or you might make one. It's up to you. But as you go into later phases and you go into commercial, you're going to make batches at a regular cadence. And again, we can scale to whatever scale we want to get, assuming we put the investment in. That's the other piece I didn't mention. And this is something that we are very serious about. Didn't mention it in the slides, but we're very serious about putting scale-up expertise in place. And this isn't trivial, but it's also not insurmountable. People have scaled up CHO cells for antibodies to 20,000 liter scale in the past. People in the cell therapy industry, myself included, have scaled up MSCs in bioreactors to 1,000 liters. Or maybe a couple hundred liters. 1,000 is more of a theoretical. So we think as long as you put the right engineering investment in place, get the right types of people, understand the technologies, we can scale this as much as we want, which will ultimately drive the cost of goods down. Greater scale, you get more economies of scale. It's just simple math. Your cost of goods is going to be lower than something that can't get to as great of a scale. And so that's one of the main advantages of this platform as well.
And then one other thing that you touched on in your overview is the analytics that you're doing in particular to evaluate the consistency and the genomic integrity of the cells as you go through the process. Can you expand on what that means and how you go about that?
Hey, Greg, maybe I want to start on one thing. I want to take the opportunity with that question, Julia, is that some people point out and that with the iPSC platform there is a risk of genetic drift. I mean, we've heard this multiple times, and that commentary reflects that whoever makes that, they don't really understand how this works. That's why I want to come back to what Greg was showing, that the population doublings are very well defined, right? And they happen primarily doing the engineering. And as I was saying earlier, at the end of the process, we screen with hundreds or even thousands, as many as we need, clones. And that's a stage where we deploy this, in-depth genetic and analytical characterization. Greg can expand, but it can go as sensitive as whole genome sequencing using short reads, long reads. It's something that you could not do in donor-derived programs where they're really highly heterogeneous populations. Here, we are starting from a single clone. So the level of sensitivity of our analytical tools is just cannot be matched, right, by in other platforms. So then, we choose the clones that objectively have retained the genomic integrity, have the right modifications. They don't have unwanted one. Those that do, because it does happen, right, with nuclease gene editing, they are eliminated. And then the master cell bank is done. And then, of course, from that, we do another, as Greg showed, there are only a handful of additional doublings, right, population doublings. And then, of course, you do another QC, but there is no genetic, there's very limited. It's just what happened within what was it that you said, Greg? A very few population doublings. So that is the risk up front, right? It's really the risk up front and subject to extensive analytical characterization.
Yeah, so just to expand on that, obviously we're looking at things like on-target integration, making sure that the gene is where we want it to be and in the right copy number. And then we're looking at off-target as well, and then we're looking at variants. So as Lalo said, we characterize the master cell bank extensively, or before the master cell bank, we characterize our clones extensively to make sure we pick the right clones. And then we characterize the master cell bank extensively because we're going to live with that for a very long time. And we look at things like structural variants with things like karyotyping and genome mapping. And then we look at small copy number variants as well. And as Lalo mentioned, whole genome sequencing is something that's really important. We're not sure where we're going to go with it. Like, for example, will it be a release test? Probably not. I think the FDA, and we have spoken about this as they probably have with many other sponsors, but when you have a clonal population, as Lalo said, you have the advantage of doing whole genome sequencing because you do believe that all your cells will be about the same. There'll be some variation, and we'll do some single cell analysis to look at that. But unlike donor-derived, where there is no clonal population, every cell is different, you really don't know what you have. You obviously got to do your best to mitigate risk, and that's in that case, but the confidence level won't be as high as it will be for a clonal population like we have. So we're going to extensively characterize with whole genome sequencing with these various tools, both the master and then final product on some regular basis and beyond final product to see what might happen. And then ultimately from that, we'll have a plan for what the release testing will look like. But right now we're still learning, and the FDA is learning with us, and they love the way we're doing this. And we're ready for phase 1. And as we learn more, we'll be ready for further phases.
Great. So you talked a lot about how your platform is differentiated from, let's say, allogeneic approaches that are donor-derived. Can you discuss how your approach might be different from others that are advancing iPS cell-based approaches, as we've seen a number advance in the space?
Well, that is hard to say. Unless our companies decided to publish detailed, protocols, it's very hard to comment, right? And it's not really, at the end of the day, we, all of us, we perform functional assays to characterize our products. And I would encourage everybody who's curious about that, to compare the functionality of the products that come out of the processes. But we don't think we can comment much more beyond that, Greg. I don't know if you want to add anything.
I'll just add one thing. I guess the next to last slide I have, which talked about process and assay characterization, and then I mentioned scale up in our chat here. I don't think traditionally, and I'm not going to mention anything about current competitors because I don't know what exactly they do, but traditionally, the cell therapy space has been very light on process and analytical technology. I saw this when I was in the MSC world, and even as I got into the autologous CAR T world in my previous experiences. And I think if you take the approach that traditional biopharmaceuticals take, which we do, and many of us are veterans of the biopharmaceutical industry, and that approach is to have a thorough understanding of your process and assays early and apply it throughout your development, and make sure you pay attention to scale up early and have a plan, because you're probably going to have to change the process as you scale up. If you have this good fundamental understanding and strong process and analytical development groups, you have a lot more chance for success. So we've invested in that. We're very serious about that. I've begun to build a group here that's more than begun. We have a great group in place that is ready to do this. And I don't know if all of our competitors can say the same. So again, I don't know exactly what's going on there, but historically people have not paid enough attention to this, and we realize the value of it.
Great, that makes a lot of sense. So I see we are at time for the session. So I'd like to thank you guys again for the great overview and discussion, and we'll be following the progress.
Thank you for the invitation.
Thanks, we appreciate it.
Thank you.